vector-borne diseases devastate crops in sub-Saharan countries, and this contributes to issues of hunger and food insecurity and limit economic development. If we are going to address these vector-borne diseases, scientists in Africa need to be able to detect the viruses and identify the insects that carry and spread these viruses. In Nairobi, Kenya, the Connected Network has brought together a cohort of early career researchers for a fully funded 5-day training program to do just this.
In my country, the labs have basic facilities which perform just basic molecular practicals. So when I saw the opportunity and the course content of what is going to be studied here, I decided to register and then join and come on board so that I can learn the new techniques in diagnostics.
I thought by coming to this training, I'll be able to meet other people from other fields, which we will be able to learn from them and also share what I know and probably come up with collaborations.
I work mainly in diagnostics, and the reason I applied for this course is because this is a diagnostic training course, and this is very important as diagnostics, they just pave a way towards more research in understanding the biology of the virus and the vector. So that's the reason why I applied for this course, and I find this course very useful, especially in my work.
Being a network of many researchers and early career researchers, we felt it would be an opportunity to network and learn from what others have actually done.
This course is important because it brings different early career researchers from different African countries all together to a single place where they can learn about the new technologies, about identifying insect vectors and also detecting the viruses in the plants or in these insects. Secondly, they will be able to apply all these learning activities in their own labs in their specific countries. During this course, we are teaching the basic molecular techniques that are used to identify insect vectors and also different and up-to-date molecular techniques that are able to detect the viruses in the plants. Nowadays, everybody is talking about the new sequencing technologies, the highly advanced molecular technologies that can be used to detect and identify the viruses. But I think it's really important that people understand the basics of detection and identification of insects and viruses. And that's exactly what we are teaching here this week in Nairobi.
So one of the diagnostics techniques that I've learned is RPA, the recombinant polymerase amplification. This is something that is new to me. I never known about it, but we have an instrument which can be used to do RPA at my institute. So through this training, the practical skills that I've learned here, I'll apply them in my work at IITA when I go back to Dar es Salaam in rapid diagnosis of brown streak and mosaic in the field.
One of the major things that I've learned is especially the methods that are used for viral diagnosis. That's one major take-home message for me that I'm going to immediately implement in our laboratory to offer diagnostic services to different stakeholders in the agricultural industry.
Experience I've gotten from the extraction of DNA from insects, I can take it back home and also apply it with my research group so that I'll teach them how to extract DNA also from insects, do gel electrophoresis and sequencing as well, and how to analyze sequence data.
One of the things that I'll actually be able to do better, first and foremost, is diagnostics, identify the diseases, because we've been doing this, but not as efficiently and as frequent. So the ability to use this tool will make it easier for us to detect it and then be able to address the challenge which we need in order to deliver clean planting materials to farmers.
So with all this knowledge will really boost my understanding, and this knowledge also I'll really pick it and take it to other students, other fellows, and also I will try and use it also in the lab.
The training workshop that connected is currently engaging with BecA-ILRI is in this mode of operation, whereby the scientists coming from all over the continent would learn the new technologies on the new methods of viruses and detections. And then when they go back home, not only they will be able to apply those the knowledge or the new method to their own program, but they will also be able to pass the information to their colleagues in the national programs. So this is where we work, and we really appreciate this partnership that we have with Connected in the UK.
If you are an entomologist like me, I really urge you to join the Connected network.
I really appreciate the Connected network for such an opportunity. It is very useful for early career researchers like me, because in here you get to meet people, you network, but also you get the practical skills.
This is a really valuable training course, and I know that Connected is arranging similar activities. So if you are an early career researcher with an interest in plant virology or plant diseases and entomology, please go to the website and register it. It's free and easy.